Biology 9700, Paper 4, Variant 1, May June 2024, question number 3 and question number 4. So, question number 3 is here. Stomata in leaves respond to changes in the environmental conditions by opening and closing. This regulates carbon dioxide uptake and water loss. Describe the mechanism by which stomata opens in the sunlight. Students, you know, these stomatas are used for exchange of gases, and secondly, the water is evaporated through them. So they are needed uh, open at the time when there is sunlight, because process of photosynthesis takes place over there. Carbon dioxide is needed for that process, and oxygen is released outside, extra oxygen, you know. So what happens during this mechanism? That is, first of all, when there is sunlight, so ATP powered protein pumps in the cell surface membrane, they actively transport the hydrogen ions out of the guard cells. So due to the movement of the hydrogen ions, so there will be the low concentration of these ions inside the cell, due to which potassium ion channels open to diffuse the potassium ions into the cell down an electrochemical gradient. So when there will be the high concentration of potassium ions inside the guard cells, it will decreases the water potential. So, to compensate for this, the water will move by the osmosis into the guard cells, and due to the entry of the guard cells, it will increases the volume of the water inside the guard cells. So they will expand, and then they will become turgid and they will be open. So we will say that these stomatas are open in this way. So the I would like to some points from this explanation. That is the um, in the in the presence of sunlight, light, the protein pumps um, protein pumps in the cell again transport hydrogen ions out of guard cells. Okay, then low concentration of hydrogen ions will be there. Ions causes potassium channel pumps to open, which allow the diffusion of which decreases the which will decrease the water tension in guard cells. So water will enter through osmosis and stomata open. So it is all about the question number A, part A. So the question number, next question is stomata. Stomatal conductance is a measure of the rate of water vapors loss from the intercellular air spaces of leaves into the atmosphere through the stomata. Our student investigated the effect of different concentrations of cyclic acid on stomatal conductance in Helianthus anus, the common sunflower. The student treated the roots of 15 sunflower plants with different concentrations of ABA solution. The plants were left in standardized condition for 24 hours. After 24 hours, the student measured the concentration of ABA in the xylem sap of each plant, the stomatal conductance of the leaves of each plant. The results are shown in figure 3.1. And students, you can see the results are shown in 3.1, and you can see the concentration of ABA is shown on the x-axis, whereas the stomatal conductance is on y-axis. So on x-axis and on y-axis, you can say the curve is decreasing. So when stomatal conductance is decreasing, when the concentration of ABA is increasing. So in this way, you can say it is decreasing between 0 to 250. It is a steep movement down in the curved, or the other, there is little change between 250 to 500. So in this way, they are inversely, we can say the inversely proportional to each other. So here we are going to write the, with reference to 3.1, decrease the relationship between the concentrations of ABA in the xylem sap and stomatal conductance. So here you can see um, as the ABA concentration is increasing, the stomatal is decreasing. So they are inversely proportional to each other. And the second point, we can write that there is steep decrease, is steep decrease in conductance to between 100 to 150 ABA concentration. So we are moving now towards the next question. And the next question is, we have got these results. And in the light of these results, we have to explain that what happened over here. So how this process has taken place, how these results can be expressed. So students, ABA or abscisic acid is a chemical which is used to close the stomatas, because when it is applied on the stomatas, the guard cells are closed. So it means ABA binds itself with the guard cells, with the uh, receptor proteins at the guard cell surface membrane. So in this way, it binds with them, and then it inhibits the proton pumps to stop hydrogen ions being pumped out. So hydrogen ions do not move out. Also, ABA also stimulate the movement of calcium ions into the cytoplasm, because the calcium ions also act as second messenger. So calcium ions, when they act as second messenger, they activate the channel proteins to open that allow negatively charged ions to leave the guard cells. When these negative ions move out of the guard cells, so it allow the movement of the potassium ions out of the cells. At the same time, calcium ions also stimulate the closure of the channel proteins that allow potassium ions to enter. So this loss of ions increase the water potential of the water. So water passes out by osmosis, the guard cells become flaccid and the stomatas close. So in this way, the stomatas are closed when the ABA acid is applied on the stomata. So I would like to write all these points. First of all, The first point, which I had told you, that is the ABA causes stomata to close. It binds. So it looks that it binds with the receptors on guard cells. So then calcium ions, it activates the calcium ions to enter guard cells, and then these calcium which act as second messenger. And second potassium ion channels open, due to which potassium ions leave guard cells, due to which the water potential is increased in guard cells. So water leaves from guard cells and stomatas becomes placid, or they close. So in this way, it takes place all about. Next question is, some transcription factors increase the rate of transcription of genes involved in the flyer of stomata. State where transcription factors bind to cause an increase in the rate of transcription. So this is the DNA promoter. DNA promoter. So moving towards the next question. Question number 4 is there. And question number 4 is, white clawed crayfish, Astoropotamius papillis, live in rivers and lakes in Europe. In 1850, the North American signal crayfish Pacifesticus lineusculus was introduced to Europe. The introduced species carried a pathogen that causes a disease known as crayfish plague. This disease kills A. papillis. Since 1850, the population size of A. papillis has reduced in many areas of Europe due to the spread of the crayfish plague. North American P. lineusculus can carry the crayfish plague pathogen without showing symptoms because they have evolved resistance to it. So resistance to it. So explain how this P. lineusculus could have evolved resistance to the crayfish plague pathogen. So you can say uh, we have to explain how the resistance is produced to the crayfish plague pathogen. So Linnaeus is that species which has produced this one. So you can say what happens at the time when they carry these pathogens. So there are some alleles which are produced as a random mutation. So these alleles will produce the resistance against this pathogen. And when there is a resistance against this pathogen, so they will multiply, they will increase their number, they will produce, and as a result of their reproduction, they have resistance against that disease. So they will produce, they will transfer these characteristics to their offsprings, to the next generations. So in this way, there will be a population of the crayfish plague pathogen, which will have resistance against this. So in this way, the resistance is produced. So I would like these in this point say so due to sometimes one or more alleles having resistance against the pathogen due to random mutation. They reproduce and transfer the genes to the next generation. Thus, they survived, they multiplied, they reproduced, and then a population is produced which has resistance against the pathogen. So next question is, it is difficult to locate uh, crayfish because they live underwater. After an outbreak of crayfish plague in one country in Europe, researchers used an environmental DNA technique to find the locations in a river where populations of A. papillis were still surviving. Water samples were taken from locations along the river. Each water sample was filtered to obtain any cells or DNA that had been released by organisms into the water. The polymerase chain reactions was carried out on this DNA using primers specific to A. papillis DNA, outline and explain the steps that occur in PCR. So we have to write down the four steps which are present in the PCR. So I would like to first of all I would like to explain them, and then I will write them over there. So in this way, the four steps which are present over there, they are first of all the denaturation. The first step, which is denaturation, it takes place at a high temperature, that is about 95 degree, around 95 degrees centigrade, in which DNA strands are separated from one another. Due to high temperature, they get separated from each other because the bond breaking takes place between them. And the second step is annealing, which takes place at lower temperature, which can be around 55 degrees centigrade. So the primer adhere to DNA strands. So primer will adhere with DNA strands. There will be the formation of DNA strands, and in the last stage what will happen, that is the which is called as extension, and the temperature is again little bit raised to 55 degrees centigrade to around 72 degree centigrade. So what happened, there will be PAQ, polymerase builds complementary strands. So in this way the polymerase chain reactions are completed in these steps. So I would like to write all these points now. So first step is denaturation, which takes place about at nearly equal to 95 degrees centigrade. DNA, in which the DNA strands are separated because due to high temperature, their bonding is break, and second is annealing. And in annealing, a little bit temperature is lowered at around 55 degrees centigrade. So what happens? Their primers adhere to DNA strands and new formation of new strands is started. And the last stage, which is called as extension, which takes place around 72 degrees centigrade. What happens over here? Track polymerase builds complementary strands. So in this way, PCR takes place. Next question is, suggest how the researchers were able to discover suitable sequence for the primer. So with the help of bioinformatics, with the help of bioinformatics, they will come to know, or they used the database, or they used make use of bioinformatics to come to know about the suitable sequence of the primers. Now, next question is, the PCR technique used in this research is quantitative. A fluorescent dye binds to the DNA and the fluorescence is monitored throughout the process, showing the quantity of DNA present. Figure 4.1 shows standard curves for four known concentrations of DNA A, B, C, and D, and the result obtained for A papilli's DNA at one location along the river dotted line. River dotted line, you can see the river dotted line over there on x-axis. PCR cycle number is there in, and on fluorescent log scale is on the y-axis, and in the center there is the threshold, and you can see the dotted line and then the A, B, C, D. State the letter of the curve with the highest starting concentration. With highest starting concentration, you can see over there, highest starting concentration, it is at A. You can see the A has got the highest starting concentration. So we will write over here the A. The quantity of A papilli's DNA reaches a threshold marked as a horizontal line or figure at 28 cycles. At 28 cycle, 28 cycle, you can see this is 28 cycle. Yes. Now, the DNA in curve C reaches the threshold after 25 cycle. After 25 cycle. So in one cycle of PCR, the concentration of DNA doubles. The concentration of DNA doubles. 25, and the DNA concentration is double. So calculate the relative difference in the starting concentration of A papillis and the DNA in curve C. DNA in curve C. In the start, it is you can say it is about what it is about This is one, and this is 2. So it becomes double standing concentration of release. So we can write it in this way. It becomes from 2, it will become 2, or in simple words 2 raised to power 3. So we can write in this way. Now the next question is, fluorescent molecules have other uses in gene technology. Figure 4.2 shows an agar plate with normal bacteria and genetically modified bacteria. The genetically modified bacteria make a green fluorescent protein. You can see the genetically modified fluorescent bacteria, and they are the normal bacteria. So it means they are modified, and how the modification takes place in them. We are moving towards the question. Identify the component that is taken up by the normal bacteria to produce the genetically modified bacteria. In figure 4.2 state what this component contains to allow the bacteria to make green fluorescent plate. Yes, bacteria, normal bacteria, and the normal bacteria, they are for these, they are called as plasmids. So they have vectors. Bacteria always act as vector because they have plasmids. Plasmids. What are plasmids? Plasmids are the circular DNA. Exact. So what happens? So they the quality of this plasmid is that it can uh, have it can we can it can bind itself with the foreign genes and change into recombinant DNA and multiply to produce the required genes. So in this way, the fluorescent proteins are formed. Suggest why the genes for green fluorescent protein is sometimes transferred in addition to the desired gene in genetic engineering, because to act as a they are as a marker gene, or to show expression of another transferred gene. So it was all about the question number 3 and question number 4. Please like, share, and subscribe my channel to get more videos, to watch more videos. Thanks for watching.